Thank you, everyone. So I will a little bit make a very quick overview of what we are doing in forensics. And of course, a lot of activities have been carried out in the frame of the IAEA CRP project. And also, I will present some case studies. So what is the role of C14 in forensics? When we talk of forensics, we usually have two requirements, which is the possibility to date very recent samples, which is not so common in C14. We have a little bit of blind period in the last couple of centuries. And well, in forensics, of course, you usually need to give good results for samples which are younger than 70 or 60 years. And of course, you want high resolution, which means a resolution which is usually not achievable for standard radiocarbon dating, which means essentially dating recent sample with a resolution of the order of 1 or a few years. So this slide has been already shown, so I will go very fast on this. We know that the nuclear detonation tests carried out after the Second World War released a huge amount of neutrons, which by interacting with the atmospheric nitrogen produced C14 at the, let's say, at the ground level, not in the troposphere as it usually happens, and which means you can see here it's a quite impressive picture for those used to to talk about C14, because also Tim was talking about the natural fluctuation of C14. C14 concentration into the atmosphere has not been constant over the centuries and the millennia, but here you see that what we have done in a very quick way, it's tremendous. I mean, in just a few years, 4 or 5 years, we almost doubled the C14 concentration into the atmosphere. And here again, the picture which have been already shown. And so I'm not going to talk too much about this. And also, I try to make this kind of calculation to explain in a more easy way what these numbers mean. The total yield in terms of explosions, nuclear detonation tests, correspond to more than 27,000 Hiroshima bombs. And of course, there are explosions by United States, USSR, and other countries, France and other countries, essentially between the middle of the 1950s to the middle of the 1960s. And these are some important aspects. The C14 concentration is, as I said, almost doubled in the 60s, but also we observed interhemispheric differences, mainly in the rising part of the curve, when the C14 concentration was rapidly changing. So, how we use this phenomenon in forensics? Now, I already talked about the atmospheric nuclear test, so you produce this red C14, which is anthropogenic C14, and then it is oxidized to carbon dioxide by chemical processes into the atmosphere, and then it becomes radioactive CO2, which is then fixed through photosynthesis essentially into the living vegetals, and in this way, through the alimentary chain, it enters all the biosphere. And so the living organisms and so on, which means that any organism is essentially does contain a trace of this anthropogenic C14. And again, if I now take a sample from this animal, for instance, in this case, it's not a case. I mean, I'm talking about ivory later on. And then if you get a tusk from this elephant, then you will have a C14 labeled samples, which means that the C14 which was produced at the ground level, you find into this animal. And then, of course, you need a little bit quite complex instrument to detect this C14 and measure its concentration. But this is the way we use this effect to date very recent sample. Then these were a little bit, if I remember well, I was showing this slide in the first meeting we had in Vienna a few years ago, showing what are the advantages and the drawbacks of what we call C14 bomb peak dating. High resolution is possible. The bomb peak curve is very well known. We have a weak resolution. I mean, we have samples taken in different parts of the world with a very high resolution in the glaciers and in both the hemispheres. So it is very well known, published, open access data. Calibration software are freely available, also with advanced statistical routines for Bayesian analysis and other way. And also, maybe Mishi was mentioning yesterday, it's a nuclear process. So this is very important in forensics. C14 is not so sensitive to environmental factors such as humidity, light exposure, or things like that. So it's quite difficult. Of course, you have contamination, of course, Irka was talking about that, but it's not that easy to change the C14 concentration. There are drawbacks, of course. I will talk later. You have multiple intercepts, and so if you measure a certain concentration of C14, there are different possibilities. Data interpretation can be complex, is not easy. Yes, Chantal Milani was mentioning this. For instance, you can, when you analyze human bones, for instance, you have to take into account the carbon turnover and fixation time. And of course, you need accelerator, and Johnny was saying this as well. So it's not an easy technique. We are getting all fast, everyone is, but also our atmosphere, which means that we are more or less crossing the 0 point, which means that if I measure the C14 concentration now into the atmosphere, it's more or less at the level of the pre-bomb value, which is not good news. This is because of the distribution of the C14 into the different terrestrial reservoir, but also because we are released huge amount of fossil CO2. And you see here that depending on the possible emission scenario of IPCC, you have different possibility of our atmosphere. Just to make it easy, it means that in about, let's say, 80 years, depending on how we'll behave or how much CO2 we will release into the atmosphere, it is possible that at the end of this century, our atmosphere will be apparently, in terms of C14 age, 2,000 or more than 2,000 years old. This is because the bomb peak will completely disappear or has already disappeared, more or less. And we are crossing now the 0 point, and it will become older, which is a limitation for the application in forensics, of course. Now, in the IAEA CRP, we had 4 worker packages, and 1 was dedicated to C14, and the main efforts were from this 3 group, the Debrecen group, Michi Molnar was representing the group, our group here at CEDAD, and also Switzerland with Irka, and we have worked very, I enjoyed very much working with them. It was very productive collaboration, and we defined also, our goal was to define common guidelines, for instance, quality assurance protocols, address ethical issues. We were discussing this yesterday, how calculate the age, and so other aspects. I will mainly say something about this intercomparison exercise, because one key point is that usually law enforcement agencies, they do not have accelerators. They cannot do this analysis by their own. They have to send samples outside, which is already a problem because of the chain of custody of the sample. But in any case, you have to show that your results are consistent, that you can measure accurately and with low uncertainty. And so we decided to concentrate on this aspect, on materials which are of interest in forensics, and again, very old samples, which are important because in this way we have an idea of the contamination we introduced during the processing and measurement. Ivory sample paper, and not going to give you many details because it has been already shown. And also this is a little bit more related to what we are doing here in a routine application, which is the use of how radiocarbon can be used in the protection against the trade of endangered species. We have a paper in Forensic Science International about this. And the problem is that, of course, the illegal trade of ivory is a relevant problem. Still, it's a relevant problem. It's causing the decline of elephant population due to poaching in different parts of the world, in Africa and in Asia as well. And because of this international convention, which is called CITES, which came into force in 1975 and was ratified by the different countries in different age, but essentially now there is a total ban of ivory trade after 1990s. This is in Italy. It may change from country to country, but around that. which means that you cannot import, buy, or even have at home an ivory sample which is younger than this age. And of course, there are different, we call them customers, let's say, that are interested in dating these samples. For instance, wildlife protection agencies, custom control services, police corps, and also private citizens, which ask us to certify that these samples have been imported or bought before this convention. So essentially the question is very easy: when was the elephant killed? And we do this by C14 dating. This is a very, because of course this international convention is protecting the living animals, not the extinct ones. So it is impossible and legal to import mammoth ivory, in Italy at least, but in several other countries. They are selling it from Russia and other countries. And so this is a problem for the protection of elephant, because they are importing ivory as mammoth ivory, and it's from elephant. And so sometimes we are asked to date. This is easy for radiocarbon. In this case, you see some of the results we obtained. These are more than 46,000 years old ivory samples, and so we are sure that this was not a living animal which was killed for getting ivory. And what we do, this is again gives you a little bit an idea of what kind of problem or complexity. Let's assume that you take one single sample from an ivory tusk, like in this case, then you go and see when in the atmosphere you had that C14 level, and as you may see, you always have two possibilities. One is before the peak, so before 1964, and the other one is after. So you always have this uncertainty because it's intrinsic in the method. What we do, we try to overcome this problem taking two samples, and such that you know the relative order of the sample. In this case, for instance, I know that the yellow point, I mean the yellow sampling point, in any case has to be younger than the red one. So I know if I am in the rising or declining part of the curve, knowing what is the growth rate of this kind of material. So I can give an estimation of the age you see with a resolution which is of the order of a very few years. So these were a little bit the outcome. This was presented at the meeting we had at the United Nations in last November, the last fall, I don't remember exactly, and you see also this was a little bit the outcome and the impact of this kind of research. And again, we had an intercomparison on ivory samples within the IAEA CRP, and again, we had very good results. I'm not going into too many details, and essentially the results were very well overlapping. And again, all the data have been published in radiocarbon. Within the IAEA, we had also a collaboration with other work packages. For instance, work package 2 was dedicated to food authentication and provenancing, and we were trying to give our contribution. And again, we were analyzing a very good sample which was given us by Johnny from Brazil. It's Melita coffee. We analyzed this. This was interesting, and I'm going to tell a little bit more about this, because when we measured the C14 concentration, at the beginning we were a little bit confused, because the C14 concentration was a little bit, you know, the sample appeared to be too old. So the C14 concentration was a little bit too high. And so when we were calibrating this against the bomb spike northern hemisphere curve, it was a little bit too old. Our data did not fit with what expected from what we could read on the label of the coffee. But then when we were writing the paper and discussing this, we realized that the C14 atmospheric concentration in Brazil and in that area is a little bit different than the average, global average. And by correcting for this, we could really have a very nice overlapping with what was expected. This slide is not updated because this is already reported in the last release of the bomb spike curve, which was published the last year. And again, by calibrating against this gives another information. when you want, or when you are looking for high resolution, somehow you can have local effect. I mean, differences between the C14 concentration because of atmospheric mixing in different parts of the world. And again, this is another sample we were also analyzing. Wine, this has been, I'm not going to say anything, has been widely discussed in the previous days of the conference, also the offset we had, and and this has been again published. This is a case studies we had. Indeed, this is a long-term collaboration with a private company in Italy. We are still asked to certify that the extracts they are selling on the European markets are not synthetic materials, but they are produced from fresh, let's say, vegetals. And you see we can clearly distinguish between, this is a way of reporting the C14 concentration, you see in the y-axis. When this is 0, it means that we are not measuring C14. When it is more than 100, it's essentially reflecting the current atmosphere. And so when you measure 0, it means that it's something derived in some way from oil or very old, million-year-old material. All the C14 has decayed. All these samples, you can certify that these are fresh material, and of course you have also the mixtures, and in this case you can calculate what is the proportion of the biofraction and the fossil fraction. And we are asked to do this routinely. Now we have a collaboration also with oil companies to assess the amount of biofuel in what they are producing. They put a very low amount, a few %, but it's important for them to have a certification method also for the commercial purposes or for several other reasons. We are measuring the CO2 released into the atmosphere from waste to energy plants. They are doing this routinely. They are forced to do this routinely because of Italian and European regulations. And they send us 1 sample per month per plant. And we are measuring what is the share of bio CO2 released into the atmosphere. This is also a little bit another application. I don't know if, okay, in this case, this is a bottle, a plastic bottle. It's on the market, and if you read the label, it says that it is produced, it's completely biodegradable, but it's produced, it's a vegetal plastic. It's 100% produced from cornstarch. And in this case, you see the importance of having or measuring both C14 and delta C13, as Lucia was mentioning, because C14 concentration gives you an indication that it is true, it's 100% fresh material, but also when you look at the C13, delta C13, and so the stable isotope ratio measured by IRMS, this is a clear indication of a C4 plant, which is corn, it's a C4 plant. So you obtain additional information. In this case also, so again, this is a certification, and there if you read in the lower right-hand side, in this case, since you do not need sample preparation, it's almost clean material. We just wash a little bit with ethanol and it's easy. You can simply drop into the elemental analyzer and you in 10 minutes obtain the C14 age and the stable isotopes. And so this is, again, a very fast method. We have also several applications in forensics anthropology. This is a little bit disappointing because we have several cases, but we are allowed to talk about a very few of them because they are open cases, or even if they are closed, we do not have the authorization to talk. This is why things can be a little bit complicated. These were samples taken from a body which was found shot in face in 2010, and we could take different kind of sample, different tissues, and different bone samples from different part of the skeleton. And again, you see when you measure or the C14 concentration, you see you have for the molar, you have again the two possibilities. But then when you measure canine, which is formed, either if it is always forming before the molar. And so in this case, you again obtain relative information which allow you to exclude this possibility. When you measure fast-turning tissue like air, you have a little bit, like Eva was talking, you have an idea of an age very close to the death of the sample, which was 2009. And of course, if you take into account the time of formation for the crown, you can give an estimation of the birth age, and by difference between the death year and the birth year, you have an estimation of the age at death. When I show this one to students, I always say that there is a part, very important part, missing in this slide, which is the error calculation, and which is very important in forensics. And here you see it's not just the instrumental error, but it's also the age on the differences. You are measuring the uncertainty you have in the time of formation, because you have very large error bars depending on the different population, the different groups, and so on. But just to make it easy, at the end you have an uncertainty on the age of death, which is of the order of ±2 years, which is not bad. But of course you need a lot of measurements to obtain this information. Again, this was published in Radiocarbon. This is my last slide. This is something I was very proud to have the permission to publish. If it will be accepted at the next Radiocarbon conference, we have one of the chairmen of the conference here, so I hope it will be accepted. Okay, this was a collaboration. We have a collaboration with the Committee on Missing Persons in Cyprus, which is a bicommunal body established at the beginning of the 1980s by the Greek and the Turkish communities, with the participation and supported by the United Nations. They are trying to identify more than 2,000 missing persons which were killed during the Civil War in the middle of the 1960s and in 1974. And we dated more than 200 individuals, because they are recovering these bones from different contexts, cemeteries, or also common graves, and so on. And they do not have, very often they do not have context information, so we are pre-screening the sample. We are saying which one of these could be, let's say, a recent sample. And for those for which we give a possible compatibility, yesterday Stefano Ridolfi was saying in Latin nihil obstat, which means it's possible that there is a possibility. Then they make further analysis, DNA analysis, they compare DNA with the database they have of the families. And then at the end, for some of them, they give the remains back to their families. And this is a very important. Now we have a new contract with them, and I'm going to present in details this. It's a little bit very touching somehow kind of study, and we are doing this. I'm going to present this at the next Radiocarbon conference, which is being organized in Zurich in the next September. And so these are very conclusion. So we showed that that C14 is really has a potential in forensics, but there are different aspects. As it was said yesterday, it's easy, but it's not simple. You still need radiocarbon expert to analyze the data, to interpret and obtain usable information. We have shown by intercomparing results, it's also very important that we didn't have any agreement about the, let's say, the processing protocol. Everyone was preparing the samples according to the standard protocols of each laboratory, and we were using different machines, and still the results are perfectly overlapping within uncertainty. And also we think that C14 can have a significant impact on real routine forensics applications. And that's it. Thank you very much. Okay, now we have it is going to be given remotely, the next talk, which is, I don't know if they need time to arrange the, yeah, maybe if you have questions in the meantime, she is already she is waiting, but they need maybe a few minutes. If you have, yeah, here she is.